Yes. So, good morning, boys and Pallavi, once again. In our previous classes, we were talking about the prokaryotic cells. We were talking about the physiology, anatomy of prokaryotic. In this session, we are going to talk about eukaryotic cells. And as I promised you, I will be putting this video in our WhatsApp group, right? You can see that. Yesterday there was some internet problem, so we could not do that. So as I said, U means clear, well defined, that can be observed. What is to be observed? Carrion. Carrion means nucleus. So eukaryotic cells are those cells in which nucleus can be seen very clearly. And it is a well-defined one. When we say well-defined, it has its nuclear membrane. Remember, we were talking about differences between prokaryotic and eukaryotic cells. And one of the fundamental differences that we talked was presence and absence of well-defined nucleus. In eukaryotic cells, very clear-cut nucleus will be present, and in prokaryotic, there will be, instead of nucleus, nucleoid. Only genetic material will be present without any boundary, without any membrane. Nuclear membrane will not be there. So it is difficult for us to identify the area of nucleus in that. Plus, we also said that along with the nucleus, other cell organelles also will not have membranes. So we said membraneless cell organelles. What are cell organelles? Parts of a cell that are present inside it, like cytoplasm, nucleus, mitochondria, plastids, all these are cell organelles. Okay. Now, you must have seen at least amoeba by now under microscope in our school, and you could have observed centrally small TT ball like structure, that is the nucleus. Okay. So nucleus is present in both plant cell as well as animal cell. Similarly, you must have observed any of the plant tissues like epidermal tissue, right, or leaf, parenchyma. So there you must observe the nucleus centrally present. And I am happy some of you are very good boys. You have drawn these two diagrams and you have put thumbs up. One or two boys are very sincere. They have put the diagram that they have drawn in our WhatsApp group. I am happy with that. And those who are lazy, don't be lazy. Draw, label it, and then don't feel that someone is not observing you. I am observing who is sincere, who is not. Okay? I have my own ways of noting down. All those who are not yet done, at least today you must do it. Tomorrow is festival, Vinayak Chaturthi, Ganapati, isn't it? So pray God and seek His blessings that you should be good in all round. Education, culture, behavior, everything, right? So when you have drawn all those good boys, and I hope Pallavi has drawn. Pallavi, did you draw?
Yes, sir.
Okay, but I am not seeing your thumbs up in the group.
Sir, labeling ready, sir.
Pardon?
Labeling I have not done, sir.
Okay, please do labeling. And while labeling, I gave two precautions. One, arrow mark should be towards the part. And since your papa is Fauzi, it should be, labeling should be in army style. Matlab, Pallavi, single file. Try to label on one side. Like you are SD, no? Senior Division NCC. So you know it when I say single file. Right? Label it. And after labeling, Pallavi, what you have to do is write the significance of each. Like when Pallavi writes ribosomes, what is the significance of ribosomes? Let us say ribosomes are removed from cell. What happens to that cell?
Protein can't be.
Proteins cannot be very good. Proteins cannot be synthesized. Raghavendra is correct. If proteins are not synthesized, Raghavendra, what happens to cell?
Can't grow.
It can't grow. So cell will be having stunted growth. Like this, you must understand each and every label's significance. Then you are perfect in the diagram. Let's say vacuoles. If vacuoles are removed, what will happen, Raghavendra?
Sir, storage waste.
Excretion cannot be there. Good, because excretory vacuoles. Then second, vacuoles also help in digestion, no? I said digestive enzymes will be there inside. So if vacuoles are removed, no digestion, no excretion, no enzymes. So the cell will have lot of problems. Like this, try to write at least 1 or 2 sentences about each label. Am I clear? At least 1 or 2 sentences about that. When you write Golgi apparatus, first you must know where is the location, where is Golgi apparatus present? In the plant cell, in the animal cell. Next, what is its duty? What is the function of Golgi apparatus? Once you know like this, you are perfect. Alright? Try to know that. Now we first go to the boundary. We have got in science school now around. Big wall is there, no? Like that. For cell also there will be boundary. And that boundary is cell membrane. And you know cell membrane is also known as plasma membrane. And plasma membrane is present both in plant cell as well as animal cell. In plant cell, it is present just below the cell wall. You have seen now in the diagram. Look at that. It is present just below the cell wall. Are you able to see? Whereas in animal cell, it is present outermost. There is no cell wall in all of us. In animal cell, there is no cell wall. That is one of the fundamental differences between plant cell and animal cell. Way back in your junior classes, you know it. So, plasma membrane is present in both plant cell as well as animal cell. Only difference is, in animal cell, it is the outermost membrane. In plant cell, it is present below the cell wall. And what is its duty? Being present outermost. What is the duty of cell membrane or plasma membrane?
Protection.
Yeah, it gives protection. Then,
Only selective objects can enter.
that's why we are calling it as selective permeable membrane. Right? Selective permeable membrane. And that means, you see, when you try to get inside science school campus, there is a chowkidar, security, in front of our gate. And the moment you come inside, they will ask your ID. Once they come to know that you are a student of science school, they will allow you inside. But if you are not so, then they will ask you a number of questions. Isn't it? Like that, this plasma membrane is like a security guard. Both incoming materials and outgoing materials are screened. And what is it made up of? It is made up of 2 substances. That is, fats and proteins. Of course, some sugar molecules also will be there, carbohydrates. So basically, the plasma membrane is made up of 3 substances: carbohydrates, proteins and lipids. Lipids means fats. And when we say lipids, which is fats, there will be 2 layers of fats. Let me expand this. Yeah, you can see here presence of 2 layers of fats, which is just like a spring. Okay, and then there is protein molecule. we have labeled it as integral protein, means in between the fat layers there is protein. Look at that lipid layers, lipid bilayer. Are you able to see? Just like ribbons there. One on top, one at the bottom. So we say bilipid layer. Bi means two, lipid. Okay? Two layers. And they are black ribbons with a background, as somebody is putting it, blue and red. Okay? Bilipid layer. And the second one is that of proteins. All these red ones, these are proteins. So proteins will be present in between the layers. So we say integral protein. Some will be present externally also. Okay? And some will have sugar molecules also. Look at that. From inside, both proteins and sugar molecules are present. So there will be two types of proteins, external and internal. But one layer only. Protein will be one layer, sugar will be one layer, but fats will be double layer. So we say bilipid layer. And that's what it says. Chemical studies on human RBC, red blood cell, show that cell membrane is composed of lipid bilayer, protein, carbohydrate. Lipids, mainly phosphoglycerides, have outer polar head and inner hydrophobic tails. Just now you saw ribbon-like structures. Those ribbon-like structures have got one head region and one stationary Kiran or Bhushan. Dhanraj is on leave, but I'll sign. Okay. You can ask Hugar madam. No? Just minute, boys. 1 minute. Textbooks is Kiran. Stationery. That's what. Stationery I am saying. Okay. Mr. Kiran or Bhushan.
Yes,
you can take. Okay, sir. So when we say bilipid layer, lipids, that is fats, they have basically they are phosphoglycerides, and they have two regions, one polar head and hydrophobic tail. Now I will expand that. I will show here. Yeah. Yes, you can see here head region and tail region. The phospholipids have got two areas. Are you able to see?
Yes, sir.
Yeah, one head region and tail region. And tail region he says hydrophobic. The other day I told hydrous means water, phobia means fear, fear of water. Remember when all of you were in 6th class, when you went to swimming pool, what was the feeling that you had first day, day 1? Were you afraid of water? Were you scared of swimming pool? But once you learnt with the help of your APTC, now you are fond of doing. That means you started liking the water, liking the swimming pool, and love the water, love the swimming. We say hydrophilic. Hydrous water, phili means love. But here the phospholipids tail is phobic, hydrophobic. Right? And I said two types of proteins will be there. Look at that. One is the peripheral protein, means outside, periphery, boundary. And another is internal. See? There are two types of integral proteins or internal proteins, means in between the lipid layer they are present. One is the carrier protein, another is the channel protein. At present you just remember proteins will be present in between the lipid layers. And two types of proteins. One is the external, another is the internal protein. Okay. Now you can see the phosphate head, which is hydrophilic, and the tail is fatty acid tail, hydrophobic. right? And you must be hearing cholesterol. Nowadays, even in your home, people will be discussing cholesterol. And generally, people have got a wrong notion. Cholesterol is bad. But being biology boys, you must tell that cholesterol, two types: good cholesterol, bad cholesterol. Good cholesterol is very essential for all of us. Okay? And bad cholesterol is not essential, and when we consume this in higher ratio, it will be causing problem for us. Hence, do not think that cholesterol itself is danger. There are two types of cholesterol. Okay? Right. Now, ratio of protein versus lipid varies in cell to cell. Like in human RBC, 52% protein, 40% lipids. Based on the ease of extraction, membrane proteins are of two types. Just now I said integral proteins and peripheral proteins. And there was a lot of discussion about the structure, anatomy of cell membrane. And we must be thankful to two scientists, Singer and Nicholson. In 1972, way back, they proposed one model for the anatomy of the plasma membrane. And that anatomy of the plasma membrane is referred as fluid mosaic model. आप ही सबमिट करना. Fluid mosaic model. Okay. It is proposed by Singer and Nicholson. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. That means there will be two layers of fat, and the proteins will be floating just like in swimming pool. So that's why the name fluid. And a mosaic, you must have heard, no? On the flooring, there will be in almost some of your houses also, the flooring will be of mosaic plates. Means lot of design will be there on the plate. So this is fluid mosaic model. Yes, please, Raghavendra.
No, sir, like a blur types and mosaic.
Blur type means some design will be there. Basically, white background plate. On that white background plate, there will be designs. Some dots will be there, some flowers will be there, like that. So these two scientists, Singer and Nicholson, they proposed this fluid mosaic model. Now, what exactly this fluid mosaic model appears? This is how it appears. Now let me expand. Yes. Look at that. From top, if you see, don't you see that it looks like a mosaic? Isn't it? Now I want you to imagine, from top, if you see this membrane, does it look like mosaic? That's why they named it as fluid mosaic model. This is most accepted version, right? And I said there are two types of proteins: peripheral proteins, which are outside, and integral proteins, which are inside, in between. You can see those blue ones even from top side when you see, right? Look at that. Look at that. So this is how the plasma membrane structure will be present. So some cholesterol molecules, also fat molecules, because we said fat will be in two layers: bilipid layer, and some cytoskeletal filaments also will be there. And you can see the presence of carbohydrates, glycolipids, that is lipid with carbohydrates attached, right? So basically our plasma membrane is made up of carbohydrates, fats, proteins. So carbohydrates are mixed with fats, so we say glycolipids. And fats, there are two layers, as we discussed, double membrane, bilipid layer, and in between proteins are embedded. So this is the structure of plasma membrane proposed by these two scientists, Singer and Nicholson. So this is the contribution of them, and this model is famous as Mosaic model of cell membrane. Clear? Now, according to this, quasi-fluid means semi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. So proteins will be moving. This ability to move within the membrane is measured as its fluidity. That's why the name fluid. And what are the functions? What is the use of the cell membrane? So basically it helps in transport of the molecules. That's why we said semi-permeable membrane, and it selects which are the minerals or elements to be allowed inside, which are the elements or membranes that are to be thrown out. Suppose some useful material like glucose is trying to escape. What happens? It filters, picks up those and puts it back. So this is how we are saying selectively permeable membrane. Suppose a bacteria wants to enter inside, dangerous bacteria, it filters out. So it's a shield, a sort of screening shield. So transport of molecules is the main thing, and it is very, very selective, right? And both inward movement as well as outward movement will be filtered. And due to fluid nature, the plasma membrane can help in cell growth, formation of intercellular junctions, means intercellular, in between two cells, right? It will form channels. That's why we saw channel proteins. And it helps in secretion and endocytosis. Endo means inside, cytosis means breaking, and of course cell division, mitosis. So this is how the protein channels, look at that. So cell membrane looks like this, and you can see the intercellular spaces. Okay, right. So extracellular space, carrier proteins, you are able to see, right? So that is the function of plasma membrane. So this is the first cell organelle that we have studied under cell organelles, and we continue this, the types. Yeah, we said cell membrane or plasma membrane helps in transport of materials. So transport means movement of materials from one area to another area. One area, let us say from Bijapur, some materials are to be transported to Hubli. So what do? Either we carry that, or we carry the materials in a lorry, or we put the materials in a goods train, isn't it? Like that, we have types of transport. Whether the material are transported through vehicle, your own vehicle, or you hire a vehicle, or through a train, goods train. Like that, here also cell membrane will have types of transport. Basically two types. One is the passive transport, another is the active transport. And as the name suggests, when we say passive transport, means without spending energy. Sometimes you must have seen, no, certain things will be moving along with the flow of water. In Krishna river, Almaty, Tungabhadra river, you must have seen Raichur boys, you must have seen Gulbarga boys, Bhima river. You just put some, drop some material in that river, just the material goes on because of the flow of water. There, it is the passive transport, means it is not spending any energy. Whereas active transport, you are spending energy. You are pushing the material, you are pushing the lorry, you are pushing the train, then it is active transport. That means in active transport, virtually you are spending energy. If energy is spent and materials are transferred from one place to another place, then it is active transport. Without spending energy, if materials are transported moved from one place to another place, place of origin to place of destination, then it is passive. Is it clear? Now, look at that passive transport. He says simple diffusion, and I think you must have studied diffusion. From high concentrated area, materials move to low concentrated area. It's called diffusion, isn't it? Like all of you must be using the naphthalene balls, white scent balls you call it, no? You just keep it 1 or 2 in your trunk or in your cupboard. After some hours, when you open your cupboard, you are having nice smell. So what is the process involved there? Diffusion, isn't it? Similarly, when you spray body perfume, after some time, your body starts giving nice fragrance. There also, the scent molecules from your body started diffusing out to environment. So that is diffusion. Here, the energy is not spent, just it is because of, I am using one word, concentration gradient. Molecules will be moving from high concentrated area to low concentrated area. This is simple diffusion. Then facilitated means here it is diffusion is being facilitated, channelized through particular way, and here there is some minimum energy will be involved. Whereas in active transport, I said there will be a lot of use of energy. Look at that, ATP is written. ATP means energy currency, I told you, right? Adenosine triphosphate. ATP means adenosine triphosphate. And I said ATP is the currency of the cell. Like Indian currency, we say rupee. American currency, dollar, like that. ATP is adenosine triphosphate. Okay? By spending ATP, if you push the molecules or materials from one source of origin to destination, then we say active transport. I hope I made it clear. Raghavendra? So, these are the two types of transports. So, movement of molecules across the membrane along the concentration gradient. What is that concentration gradient? High concentration to low concentration, without the expenditure of energy. Then we say passive transport. And in that too, simple diffusion, movement of neutral solutes across the membrane. And second one is osmosis, movement of water by diffusion across the membrane. But you must know the difference between diffusion and osmosis. I hope in junior classes you have studied. Diffusion is the movement of solid particles from high concentration to low concentration, whereas osmosis is movement of liquid particles as well as solid particles. But one condition is there. What is the condition? Across the membrane, there should be a membrane, plasma membrane should be there. And you can define osmosis in through two ways. I think we will study that separately, right? You can see the same diagram again, passive and active transport, and these two are done through osmosis. So polar molecules cannot pass through non-polar. Polar means there will be charges, polarity, you must have heard, no? Positive and negative charges. So polar molecules cannot pass through non-polar lipid bilayer. So they require a carrier protein for the transport. If materials are transported through that carrier proteins, then we say facilitated diffusion, right? And further we can see here the types of the transport movement of molecules across the membrane against the concentration gradient from low to high concentration with the expenditure of energy. Example, sodium plus and potassium plus pump. Now, what happened here? If movement molecules are moving from high concentration to low concentration, no problem, because concentration gradient is there. Without spending any amount of energy, materials will move. But what happens if there is movement from low to high? Already materials are low, and from low they have to move to high concentration. Then there is requirement of energy to be spent. ATP molecules are to be spent, right? Then we say, by spending energy, if materials are made to move, then it is active transport. I hope I made it clear. Active and passive. And tomorrow we will see cell wall, structure of cell wall, right? Take care.